Hi everyone, welcome back. We're going to finish the cycads today. I am here in Brooklyn in a basement, so hopefully the sound is okay. I've been having technical difficulties while I've been trying to do this, so this is like the third time, so hopefully this works because it's been frustrating trying to do this. We're going to finish up the cycads today, and where we left off last time was the pollen grain, and we kind of got all the way through to the pollen grain, but I want to kind of back up a little bit and just remind ourselves where we are even at in the cycads and a little of these terms that we came across last time about cycad reproduction. So last class, cycad reproduction, so we are in the seed plants, right? We're in the seed plants, but we're in the gymnosperms. So gymnosperms have naked seeds, and that means that gymnosperms have naked ovules. So cycads are a type of gymnosperm, so they're going to have naked ovules and then later naked seeds, right? So we covered this last time. This diagram is showing you a picture of an ovule. Inside of each ovule, you'll have a megasporangium that will have one functional megaspore. And that megaspore will grow into the female gametophyte plant, who will live inside of the ovule. Female gametophyte plant will end up having all the things a female gametophyte plant tends to have, meaning sex organs that make eggs, right? So the reason why the ovule is such a great thing in the evolution of plants is because once the female gametophyte plant, so once this functional megaspore grows via mitosis and cytokinesis and turns into the female gametophyte plant, which will be inside of here, right? She'll have on her archegonia that have eggs in them, one egg per archegonium. And the female gametophyte with her eggs will be better protected because she's inside this ovule, and the ovule has layers called the integument that surround the female gametophyte plant who lives inside of this megaspore. And then eventually sperm via the pollen grain, which is where we're going to get to in a few minutes. Pollen grains will be able to enter through this hole called a micropyle, and they will bring the sperm to the egg inside. So microsporangium, so this is where the megasporangium are, or this is one megasporangium, right? And it's inside of an ovule. Microsporangia are not inside of anything. The microsporangia are just on specialized leaves, which we Microscorophylls, which produce, but the microsporangia still do what we're used to, in that they just produce microspores, right? Same thing. And I said last time that these little yellow doodads here are each one of those is a microsporangium, and they kind of almost look like a little Pac-Man. All right. The ovules are attached to leaves called Microsporophylls. The microsporangia are attached to leaves called Microsporophylls. These are terms that we first came across in Selaginella lecture. So the Microsporophylls are sometimes, get it together, me. The Microsporophylls that have the ovules on them are sometimes arranged in a Strobilus, sometimes not. As we said last time, that sometimes they're in a Strobilus, sometimes they're in a fertile crown, right? A crown of fertile leaves. But the Microsporophylls that have the microsporangia on them are always going to be arranged in a Strobilus. So here on the left, we have examples of the male and female Strobilus. Here is a male Strobilus. This is a female Strobilus. So in this species on the left, sorry, my husband's roommate is watching basketball. So you might hear some yelling periodically. So the sporangia, the Strobilus on the left is the male Strobilus, and always Microsporophylls are going to be arranged in a male Strobilus. But like I said, Megasporophylls are not always arranged in a Strobilus, but in this species they are, right? So here, this is female Strobilus and a male Strobilus from the same species. There's a lot of diversity in what this Strobilus can look like, but ultimately they all contain the same things, which is a stem, leaves, and sporangia. All right, how do I do this? Okay. So, like I said, the strobili can vary a lot in shape, size, and color. This is just showing you, and this is all review from what we did last time, but I just kind of want to work up to the pollen grain. So cycad plants are strictly dioecious, meaning they have two houses. So we always, in the cycads, we always have female plants and male plants. Female meaning that they have, one plant is going to make megaspores ultimately, or shed, one plant is going to be where the megaspores are found, and the other plants are going to be where microspores are found. In the female plants, the Microsporophylls are, like I said, either in a crown of fertile leaves or in a Strobilus, depends on the species. So here on the bottom, it's two examples from different species. In the one on the left is the crown of fertile leaves, and the one on the right is the Strobilus. So both of those have Microsporophylls, but they're just arranged in different ways depending on the species. Remember, cycads don't have cones. So the book may say cone, you might see cone online. Only conifers have cones. Cycads don't have cones, they have strobili. I pointed this out last time. So this whole thing on the black is not a good color to use on this slide. This whole thing here is the Strobilus this is a female Strobilus. It consists of Microsporophylls, right? So special leaves, and on each of those special leaves is going to be an ovule, right? And so you can see the ovules here in red. This is the thing I said before, that these leaves almost look like oyster crackers, like the crackers that you put in soup, right? And then a real thing on the right here. So this is an example of when Microsporophylls are arranged in a Strobilus. This is an example of when Microsporophylls are arranged in a crown of fertile leaves. And like I said last time, the leaves that are in the Strobilus don't really look like leaves at all. They kind of look like weird cracker things. Whereas the leaves that are in these crown of fertile leaves look a little bit more like a leaf, right? Look a little bit more like a leaf. But regardless, if you're a Microsporophyll, you have an ovule attached to you. So whether you look like this weird little kind of soup cracker thing, you still have an ovule attached to each Microsporophyll. And likewise, if you're this leaf in the fertile crown, you'll still have ovules attached to you as well. This is what I showed you last time. The leaves of the fertile crown look more like leaves compared to the leaves of the Strobilus. But overall, there's just a lot of diversity in Microsporophyll shape. And the number of ovules per Microsporophyll will depend on the species. So this one on the right has 2 ovules, whereas the one in the middle has 6 ovules, etc. Depends on the species. Because cycads are strictly dioecious, that means we have what? Male and female plants. And the male plants are going to make microspores. And the Microsporophylls are always going to be arranged in strobili. So it doesn't matter what species you look at, it's always going to be arranged in strobili. So this is a male strobili shown here on the left. Keep going back to this here. Let's pick bright green. So this is a male Strobilus coming off of this tree. And if we were to look closely at the male Strobilus, you would see a bunch of Microsporophylls, right, which are leaves that have Microsporangia on them. If you were to pull one of these Microsporophylls off, it would look like this. And on the bottom side of that Microsporophyll, you would see all these little dots, which are each the Microsporangium. Here is the same thing. This is a real leaf here on the right compared to a drawing. And you can see the little, I think I said fish egg looking things on the bottom of that leaf are the Microsporangium. And this is kind of getting to where we left off last time. In that, now that we're in the seed plants, the male gametophyte plant is even more reduced. So there's this theme throughout plant kingdom course where the gametophyte plants are getting more and more and more reduced over evolutionary time. And the sporophyte plants are getting bigger and bigger and more complex over evolutionary time. And so this is one of those pivot points where we see that reduction of in this case, the male gametophyte plant. So if you don't remember back with Selaginella, where we first learned about that was the first time that we ever came across sporophyte plants that were heterosporous, right? Was a Selaginella. And remember when we had, so heterosporous meaning the sporophyte plants made two different types of spores, megaspores and microspores, right? Well, remember, and so if you have megaspores and microspores, it means that that also means that the gametophyte plant is now going to live inside the spore. So that might be a good way to kind of organize things for you. Know that if the sporophyte plant is heterosporous, the gametophyte plants are always going to live inside the spore. So if you have two types of spores, you're going to have two types of spore, each of which has a gametophyte plant inside, a male or female. But just, I'm getting distracted, but back in Selaginella, remember, here's our microspore of Selaginella. What was inside of the microspore in Selaginella? It was a single antheridium. So already, remember, Selaginella is what? A lycopod, right? And what is also a lycopod? Lycopodium. Lycopodium is homosporous, and so its gametophyte plants grew outside in the world, right? Like all the non-vascular plants. But by the time we get to Selaginella, our first example of heterosporous sporophyte plants, we already see a big reduction in size of the gametophyte plant, in that inside of the microspore, the male gametophyte plant had been reduced to a single antheridium. That's it. Remember, I think I said Dr. Casper used to call it testicles in space because it's kind of what it is, right? You have this microspore floating around, and what's inside of it? An antheridium, right? It's a male sex organ. So that's why you call it that. So that's already a big reduction. But now that we're in the seed plants, in particular cycads, but in the seed plants, the male gametophyte plant is going to be even more reduced. And this is where we kind of left off last time. That's why I was going a little fast through that. That we're going to have this new evolutionary leap, right? Oh, I got to move all this stuff here. Big new evolutionary leap, the pollen grain. Pollen grain. So inside, and I'll show you on pictures here, but inside of the microsporangia. So where is that again? Where does that look like again? That's like here. So if you were to pick up a male Strobilus and pull a microsporophyll or a leaf off of that Strobilus and look underneath the leaf, you'd see these little fish egg looking things that are each microsporangia, right? So inside each of those little fish egg looking things, inside of those are microsporocytes, or you can say microspore mother cells, same thing. But the main thing about it is that they are diploid, right? So there's cells inside of the microsporangia that are diploid, right? You can call them microsporocytes. And those diploid cells are going to undergo meiosis and cytokinesis to make a tetrad of microspores. That's not different than anything else we've seen so far when it comes to spores being made, right? 2N cell inside of a sporangia undergoes meiosis and cytokinesis, and you end up with haploid spores, right? That's the same as we've seen for everything so far. But okay, we have heterosporous spores, so you have micro, right? Fine, but same old thing. Now we're going to have another step. So each of these microspores is going to go through another process in that they're going to go through, look here, the microspores are going to undergo mitosis and cytokinesis, meaning if they're going to grow, if you see mitosis, you think grow. And as that happens, each microspore is going to grow into what's called the pollen grain. And the pollen grain is now the very reduced male gametophyte plant. That's what the pollen grain is. How does that happen? Okay, let's go through it. Oops. Oh, and this is something I said last time. These are the parts of a pollen grain. Pollen grain has a prothallal cell, which we don't really know what it does. There's a pollen tube cell that allows the pollen, the sperm inside of the pollen grain to grow to the egg. And there's a generative cell that generates the two swimming sperm. So these are the parts of the pollen grain. All right. So here is what a pollen grain would look like under the microscope. This is Cycad, oh, sorry, this is Cycad sperm, not pollen grain. So this is the sperm that's inside of the pollen grain. And just like the more primitive plants, the cycad sperm are able to swim. You might see they have all these little like tails here. But they don't swim through water. Instead, now they're swimming down the pollen tube. All right. So let's go through this real quick to just kind of look at how we get from a microspore to a pollen grain. So this is the microspore that would have resulted from meiosis, right? Remember, right? You're inside of the microsporangia. There are diploid cells there. They're going to undergo meiosis and they're going to make microspores, right? So now on this slide, oops, on this slide here, this is here's one microspore, single cell microspore. It's going to undergo mitosis. This is what I just said in words on the previous, but I'm showing it to you in pictures. The microspore is going to undergo mitosis and cytokinesis. And the result will be pollen grain. So you could see here, the microspore is just a single cell with a nucleus inside, right? After it undergoes mitosis and cytokinesis, now it's technically two cells. So you got this cell here, oops, and this cell here. And when it's finished going through these mitotic divisions, this is what you'll end up with, which is technically 3 cells. But 2 of the cells are inside of one of the cells. So the whole thing, this whole thing is the pollen tube cell. This little thing here, maybe you should use different colors. So the green circle is the pollen tube cell. This cell inside, this red thing here, is the generative cell that's going to generate the 2 sperm. And then this cell on the bottom here is the prothallal cell that we really don't know what it does. And it may just be left over from when there used to be a prothallus. So eventually this pollen grain, this whole thing, is going to get released from the microsporangia and land on an ovule. And when that happens, this pollen tube cell, which remember I said is the whole outside is the pollen tube cell, is going to elongate and grow down to meet the ovule and allow the sperm to swim. And so that's what you see here at the bottom. This is the pollen tube cell that's growing. So this is extending down, growing down to meet the ovule and bringing the 2 swimming sperm with it that swim down the pollen tube, right? So there you go, pollen tube and the 2 sperm that swim. Here's what the sperm look like close up. You can see all of that cilia, which is kind of like little flappy things that help it swim. And so when I was going through this here, I said that, remember, eventually this this whole thing, right? So this is the pollen grain. This whole thing is the pollen grain. All of these components: pollen tube cell, generative cell, prothalial cell. Eventually, the pollen grain is going to get released from the microsporangia. So because of that, you can call them, in the seed plants, you can call the microsporangia a pollen sac, because what they release now is not a microspore. They instead release a pollen grain. So I think there's a question in one of your, I think it's in your lab. I can't remember, but basically this microspore here, this microspore here won't ever leave the microsporangia. It will stay inside the microsporangia and undergo this process to become a pollen grain. And it is pollen grain that is ultimately what gets released from the microsporangia. So AKA pollen sac, because now the microsporangia make pollen. So there, that little Pac-Man, you can call them a microsporangia. You could also call them a pollen sac because it releases pollen. All right, how are we doing here? All right, we're gonna just briefly, now we know all the players of cycad reproduction, so we're just gonna go through alternation of generations to put all those players together. So remember that cycads are strictly dioecious. So you have female sporophyte plants and male sporophyte plants. Don't get confused. So like, remember, it's the sporophyte plants are making spores, right? And the spores that the female sporophyte plant makes is megaspore. And inside the megaspore is the female gametophyte plant. So just don't get it like twisted. It's not that the sporophyte plants make gametes. They don't, but they make these spores that give rise to those gametes, right? Write me a question in the discussion if that, what I just said right then, was confusing, because I could see how it would be confusing. But sporophyte plants make spores. Anyways, cycads, strictly dioecious in that they have female plants and male plants, right? So these are our sporophyte plants. How do we know they're sporophyte plants? Because they are diploid, right? Diploid sporophyte plants. Not palms. All right. So first, we'll talk about this female sporophyte plant on the left here. So what is on the female sporophyte plant? Well, for this species, it has strobili. Remember, other species have the female Microsporophylls arranged in a fertile crown, but this species here has strobili. So on the left here, this picture on the left is the female Strobilus, which is diploid because it's part of that sporophyte plant, right? So we have female Strobilus, is this whole thing on the left. And on that Strobilus, what is a Strobilus made out of? What is a Strobilus? It's a stem with leaves that have sporangia on it. That's what a Strobilus is. So on this Strobilus, we have leaves that, in this case, they look like those little oyster cracker things. Those leaves, we call them Microsporophylls because they have Microsporangia on them. However, now since we're in the seed plants, what it really has on it is ovules with Microsporangia inside. So if we were to look at one of these leaves up close, that's what this is. And you can see that on this species, this leaf has 2 ovules on it. Each of these is an ovule. And inside of that ovule would be the female gametophyte plant. So there's the female stuff. This is just saying top and bottom. I don't really care so much about ad or avaxial. Here's what's on the male plant, right? Because we're strictly dioecious. Male plants and male plants. Female plant has ovules on it. Male plant has microsporangia on it, aka pollen sacs. So if we were to look at this, this is the whole, whoops. This is the whole what's a good color? This is the male Strobilus, right? So it has a stem and leaves on it, and the leaves have sporangia on them. Because we have male or microspores are going to be made, we have microsporophylls, that's what the leaves are called, and each microsporophyll will have a bunch of microsporangia on it, aka pollen sacs. Here is one leaf, right? One microsporophyll. Here is one pollen sac, right? The little fish egg looking things. Okay. All right. What's going on inside this ovule, right? What's going on? Well, we already learned a little bit. So we know the ovule has inside of it a megasporangium, and that the megasporangium has one functional megaspore. This down here says Microsporocytes. It's the same thing, but it has one functional megaspore. The megaspore, which is here in this drawing, which is diploid, has to undergo meiosis and cytokinesis. Oh, sorry. Yes, sorry, my bad. In the ovule, you have a megasporangium. In the megasporangium, you have diploid cells, like spore mother cells, that undergo meiosis and cytokinesis and result in one functional megaspore. That functional megaspore is going to undergo mitosis and cytokinesis and grow into the female gametophyte until this whole thing is just full of female gametophyte, right? That I'm drawing in green. So, so that's what's going on inside the ovule. What's going on inside of the microsporangia, aka pollen sac? Well, we just learned it, right? Inside each pollen sac or microsporangium, there are microspores. No, gotta take a step back. It's the same as before, right? So in each microsporangium, which is the fish egg looking thing, there are microspore mother cells or microsporocytes that are 2N. Those are going to undergo meiosis and cytokinesis to make a microspores. Here is one of them. Here's one microspore up here. That microspore is going to grow through mitosis and cytokinesis into the pollen grain, which is now the highly reduced male gametophyte plant. And you might think to yourself, what do I mean by highly reduced? Well, in Selengenella, as I said at the top of the class as a reminder, the male gametophyte plant was reduced to a single antheridium, right? Which is very small. Here, what's the male gametophyte plant reduced to? Basically 3 cells. 3 cells: pollen tube cell, generative cell, and prothalial cell, right? 3 cells. That's a big difference of one organ like antheridium versus 3 cells. 3 cells are way smaller than an organ, right? So that's why we say highly reduced male gametophyte plant. Okay, how are these sperm going to get to the egg, right? Because we know if a female gametophyte plant is inside of the ovule, right? I don't know why I keep doing that. I think I just like doing that. We know female gametophyte plant is going to end up inside that ovule, and so she's going to have an egg. And we know that the male gametophyte plant has been reduced to this highly reduced thing called a pollen grain, which is basically a 3-celled male gametophyte plant. Part of that has sperm. So how are the sperm going to get to the egg inside the ovule? Here's how. So first, this female gametophyte plant, she's going to grow inside the ovule. This, I guess, is why I keep using the green color, because of this slide. So now in this ovule, it already contains a female gametophyte plant. It's already made. And you can see she has her archegonia on her. She has 2. It says that at the bottom here. And I've color coded this so for your convenience. So when you're going through the slides, it's a little more intuitive. So female gametophyte shown in green here, she's inside of that ovule. And there's 2 archegonia on her. Oh, and notice, just to make it clear here, look, I shouldn't use red. The ovule is 2N, right? Why? Because the ovule is part of the sporophyte plant. Just let me go back here. Right? Right? Here's a female sporophyte plant, right? On the left, on that female sporophyte plant are strobili with leaves, and the leaves have ovules on them. So the ovule is part of the sporophyte plant. But inside the ovule is that Microsporangium where the female gametophyte will grow. So I'm pointing that out because look here, female gametophyte plant, which is shown in green, is inside the ovule, but the ovule is diploid because the ovule is part of the sporophyte plant. But the female gametophyte plant, she's haploid, right? So anything on her is haploid. So her 2 archegonia shown in blue, those are haploid, and each egg inside of each archegonia shown in red is also haploid, right? Because those are all parts of the female gametophyte plant. So that's all inside the ovule. Just to make it clearer what we're looking at here. So there's that same thing, female gametophyte plant shown in green. And she's really, really, really well protected. How is she well protected? She's got, first of all, the Microsporangium, aka nucellus, surrounding her. And on top of that, she has these many layers of the integument surrounding her too. So look how awesome these eggs are protected, right? Compared to plants we've looked at in the past. Much better protection for that egg, ultimately much better protection for the embryo once eggs fertilized. Okay, here's our good friend pollen sacs, right? Looks like little Pac-Man. So once the pollen grain, remember this, once the pollen grains are ready, Microsporangium will open. So this is them, sorry, I'm not trying to make you dizzy. This is what the microsporangia look like when they are closed, but when they're finished making pollen grains, they're going to open up like little Pac-Men. Ah, there you go. Well, right, they're open. And release the pollen grains. Remember, we're going through this to say, how does the sperm from the pollen grain end up in fertilizing the egg? Do I color code this? No. So pollen grains are going to get released from the microsporangia, aka pollen sacs. As the pollen grains are released, eventually they're going to land on an ovule. So, and there's different ways that this can happen. Sometimes insects can bring them, sometimes wind. It depends on what species you're talking about. But, so this whole thing is an ovule. And what happens is the ovule, through the micropyle, remember the hole, a little sticky drop, which is called a pollination drop, will come out of the ovule at the micropyle. And what'll happen is the pollen grains, which I'll do in, I guess, orange, they will land on this sticky drop. And then eventually the sticky drop is going to kind of dry back up into the ovule, and it will go back in and bring with it the pollen grains into the ovule. That's what this says down here. When the pollen grain lands on the ovule, we call that pollination. So pollination is when the pollen grain lands on an ovule and lands on, in this case, the pollination drop. Okay, so here you go. Remember, this is where the pollination drop would have come out, this micropyle. So here, I'm going to use the same color I used before. So pollination drop would have been coming out of here. Pollen grains landed on the pollination drop. And then eventually the pollination drop's going to dry up and go back into the ovule, and that's going to bring the pollen grains with it. See, that's what it says on the bottom. The first place that the pollen will enter the ovule is called the pollen chamber, aptly named pollen chamber. Notice the pollen chamber is not yet, let's use purple, not yet inside of the wall of the Microsporangium. So here's the Microsporangium here in purple, or the nucellus. Notice the pollen chamber is still outside of that. So the pollen grains at this point are inside of the ovule, but they're not inside of the Microsporangium yet. And they have to get in there because that's where the female gametophyte is. So they enter the pollen chamber, and the pollen grains in the pollen chamber, they'll land on the outside of the Microsporangium, right? Purple thing here. They'll land on the outside of the Microsporangium, and the pollen tube cell will grow. So look, so here on the left, you have the pollen. I'll do it in orange. So here is a pollen grain that was brought into the ovule via the pollination drop that would have been coming out of the micropyle. Pollen grain, shown in orange here, is now inside. Where is it? In the pollen chamber. And then the pollen tube cell is going to start to elongate and grow into the wall of the Microsporangium. See that? Into the wall of the Microsporangium. This is very complex, right, guys? Like, this is a very complex thing compared to what we've seen before in plants. So the pollen tube cell is going to grow, grow, grow. As it grows through the wall of the Microsporangium, the two sperm will swim down the pollen tube as it grows. Okay. Now this pollen tube cell has grown through the wall of the Microsporangium. So look, pollen tube reaches the inside of the Microsporangium, and the swimming sperm enter what's called the archegonial chamber. So now they are officially inside of the Microsporangium. So just to get you situated, and I think your lab has this exact same diagram or something very close to it. But oops, you have the micropyle, which is the hole. The pollen grains enter through the micropyle via the pollination drop. Once they get inside of the ovule, they're in the pollen chamber. Once in the pollen chamber, the pollen tube cell will grow through the wall of the Microsporangium and allow the two sperm to enter the archegonial chamber shown here. And so here is where the archegonial chamber is located with respect to the micropyle and the pollen chamber. And then that's going to allow the sperm to be close enough in proximity to the eggs that are in each of the archegonium. And the sperm, the two sperm will fertilize an egg. I'm sorry, the two sperm enter, but only one sperm wins. Only one sperm wins. So one sperm will fuse with one egg. So it's kind of almost a race. Like, you have these two sperm and you have two eggs. See here, I'll use pink. Here's a sperm, here's a sperm, right? They're swimming down the pollen tube cell. Here's an egg, here's an egg. So there's two sperm, two eggs. One sperm will win. One sperm will win. So let's say this sperm fertilizes this egg. That's the winner. This other sperm, sorry guy, it'll just degrade, and this egg, sorry egg, will just degrade. So it's kind of a race to see who wins. But when the sperm fuses with the egg, that's the process we're already used to, which is fertilization, right? The other sperm will degrade and the other egg will go unfertilized. And it can take months. I want to make this very clear. Months between the pollination event and fertilization event to occur in cycads. So I don't know if you remember last time in the cycads part 1, I said that cycads are facing, many species are facing extinction. This is part of the reason why, because it can take months to go from pollen grain landing on an ovule to egg and sperm fusing. So just to get you like, remember this? So pollen grain lands on the pollination drop. And then months and months and months go by where all these processes are happening. Months go by until finally fertilization happens, right? So that's part of the reason why cycads are facing extinction. So obviously when you have egg and sperm, haploid egg, haploid sperm come together, it results as a 2N zygote. And the zygote for cycads is actually going to undergo lots and lots of mitosis without cytokinesis. So this is why at the very beginning of the course, I made a big deal of saying, you know, you have mitosis and cytokinesis, because you can have mitosis many times in a row without cytokinesis. You can have mitosis, then cytokinesis, or you can have mitosis, cytokinesis. But in this case, once the zygote gets fertilized, there's going to be a lot of different mitotic divisions. So mitosis is going to occur lots of times until eventually the cell membranes form and you have multiple cells. This is also what happens in, for example, fruit flies and other animals. And it has to do with body arrangement, body plan stuff that we don't cover in this class. But in this diagram here, here's a fertilized egg. Here's many mitotic events occurring, but no cytokinesis. All of these new nuclei, the result of all these divisions, or sorry, the result of all these mitotic divisions or all these steps of mitosis, you get all these different little red nuclei here. Eventually the nuclei kind of get organized, and then finally, as shown here, the cell membranes start to form around all of them. It's that. Anyways, just to show you why I say, you know, mitosis and cytokinesis instead of just part of the same thing, right? So cytokinesis is happening there at the end. Yes, now we have an embryo. And the embryo is going to be what grows inside of the seed. So, all right, so let's look at that. So here, we've already had fertilization occur. We've already had the zygote turn into an embryo. And now we're inside of the seed, right? Because the ovule will turn into the seed once it's fertilized. So this here, see here it says nucellus. So that's the wall of the Microsporangium, what was the Microsporangium, and then the integuments would be beyond that. Anyways, now though we're not an ovule anymore, now we're a seed. So this will become the seed coat. We'll learn more about that. But first, the embryo. So what does an embryo have? So just so you know, this thing in the middle here is an embryo inside of a seed. The embryo has baby leaves, which we call cotyledons. So here's a baby leaf. Here's a baby leaf. The embryo has an apical meristem because it's going to need to grow in length. And so it's going to be doing primary growth. So the apical meristem, I'll use a different color for apical meristem. Apical meristem is here. Then there is going to be a baby root, embryonic root called a radicle. The radicle root cap is here. And then there's a stem area that's between the leaves and the baby roots. We call that stem area a hypocoidal. So that's here. And just so you know, hypo means below, and coidal refers to cotyledons. So the hypocoidal is basically saying it's the stem tissue below the cotyledons, right? And indeed it is, right? Because where's the hypocoidal? Below the cotyledons, right there. Radicle here, cotyledons here and here. Okay. Woo-wee, here is a picture of it in real, just to give you an idea. And what's going to happen is as this embryo grows, it's going to need a food source. And so actually, which is a little kind of macabre, but so it'll eat the female gametophyte plant. So it's like, thanks, Mom. Yeah, so the embryonic plant will consume the female gametophyte plant as nutrients so that it can grow into the new sporophyte plant. Here is what seeds look like on cycad. Sometimes they can be very diverse, but these big red things here are the seeds. Notice the seeds are not inside of any fruit or anything because we are in the gymnosperms, which are naked seeds. Believe it or not, these weird yellow things are actually Microscorocytes. They're leaves, believe it or not. But the big red things are the seeds. Okay. So let's take one of these seeds. The seed falls off. Inside of the seed is what? The embryo, right? The baby plant. So as the embryo is going to keep growing, and eventually the root, so eventually like the radicle root will come out, and then eventually the shoot and leaves will come out, and then you'll get brand new, then your sporophyte plant will grow. And here is where we kind of started. New sporophyte plant will emerge from the seed. Okay. It's a lot of material. Definitely it's going to take time to parse this apart. So I'm glad that we at least spread it over 2 lectures so that you guys can, you had some of it last time, we're able to sit and marinate on it a little bit, finish it up today. And so kind of break it up as versus one giant lecture, right? Okay, so I was gonna try to start ginkgo today because technically in class right now, we still have a half hour. Yeah, we still have a half hour. So I think I am gonna start it just to keep us on track. Should I start it? No, I'm not gonna. It's too much material for one class. We'll do ginkgo on Thursday. But regardless, so now today you have finished cycads. And so a week from today, you'll have an assignment due that covers the introduction to the ovule, like the amazing ovule lecture, and the cycad lecture. So that's due in 1 week and it's available now on Canvas. And then on Thursday, we'll cover ginkgo. Okay, cool. So with that, thanks so much, guys. And many of you have reached out to me and met with me for office hours. So I really am thankful that you've done that. Please, if you aren't doing so well in the class and you're not sure why, or if there's questions you have about some of the material I've covered or anything like that, just please reach out to me so we can schedule office hours. I feel like students tend to find the office hours pretty good. You could ask your peers. But anyways, yeah, I'm always here to help. So send me an email if you want to do office hours. All right, bye.